So, there are two main bits of this talk. I'm the boring part, I'm afraid. So I'm doing just a crash course in the basic concepts and techniques behind modern molecular biology. So roughly...
Way!
Thank you. I should say, actually, most of my time I spent in front of crowds. When I was a teenager, I used to do kids' parties like magic and balloons and circus and things. So I am used to a very responsive audience. Please do feel free to ask questions, heckle, and ideally throw things at Sam. Because, there's always one unpopular kid in the crowd. I'm sorry, Sam. Equally, if another hangover from doing parties, if I start to look panicked and start reaching under the table, it's because I'm looking for sweets to throw into the crowd to appease you. But sadly, no.
Appease us!
Yeah, dear, what do you want?
I don't know.
Can you make a poodle?
Out of...
Balloons.
Oh yeah,
I'll get right on
Make a
that.
box.
Yes.
I can make a poodle.
So as I said, yes, I'm doing the relatively boring part. I'm enough of a geek, but I actually think this stuff is really cool. So if I go to glazed eyes and off on a massive tangent, please, again, heckle, slow me down. Equally, if I'm going too fast, because I have no idea what level you guys are at, stop me, ask questions, and make me explain myself. The interesting part of the talk, I think, is Sarah. So she is, well...
I don't have the same expertise on balloons and parties,
but Sarah knows much more about the actual biohacking scene than I do. So what projects people are actually getting up to and the problems faced and so on. And so I'm the sort of sprout, Sarah is the media actual man. So yeah, we'll get on with it. So, if this works... Oh, come on. Oh, you suck.
This is not going to get old at all.
Can somebody just press it?
Yeah, do you want me to do it for you? And I'm sat here. Machine hang. Running Windows.
Open them.
No. This is why you shouldn't use open source, people. It's rubbish. Thanks for that. Some people are actually doing statistical biology as well, so mathematicians with exactly the right amount of time on their hands, who are taking massive data sets online that professional researchers don't have the time, inclination or math skills to work on themselves, and either double-checking published research they don't believe, or doing some quite interesting novel bits of research. There's also the physical and chemical research and engineering stuff, which is actually building the lab equipment that we need in order to... Yes. That we need in order to run a lab. Because bits of lab equipment, it's all available, a lot of it's available commercially, and most of it's horribly expensive. But there are nice workarounds available. So, next bit. So, moving on to the introduction to biology itself. This will bore some of you, hopefully not all, but it's an essential bit of background for sort of to understand biohacking. So there's what's called the central dogma of molecular biology. And the idea is that the flow of information goes from DNA to RNA to protein, which does something. And that's a basic fundamental idea. So DNA in itself doesn't do all that much. RNA is a sort of a messenger, if you like, that carries the information away from the DNA, acts as a template to make protein, and the protein does something interesting, one hopes. Or, in my experiment last week, gathers in little clumps and makes me shout rude things at the machine.
Something.
Yeah, well... so, next. So, starting with the DNA. Information is encoded in our DNA. The molecule itself is chemically moderately interesting, depends which chemist you ask. Not terribly complex, though. So you have 4 bases, 4 letters, A, C, T, and G. And they comprise, essentially, recipes for manufacturing proteins, which do all the work in the cell. So you have some combinations of letters act as binding sites for proteins to come and stick to the DNA and start copying off bits of RNA. Other sort of stretches of DNA encode sort of stop signals, so it goes at the end of a gene. So you have a binding site, a start signal, the gene itself, then a stop signal. And there are other bits and pieces. You get some targets for modification of the DNA as well, which I'll mention a little bit later on. So you can see what's called a karyotype. So if you take all the DNA out of one cell, and you can see our chromosomes down in the bottom corner over there, we have that many chromosomes.
46.
Thank you. I'm saying I'm going to fire my boss ever sees this. But together, there are about... So this is what we call diploids. You have 2 copies of each one. When you have 1 copy of each one, there are something in the region of between 3 and 4 billion bases encoding something between 20 to 25 protein-coding genes. Now that seems like an awfully low ratio, and it is. A great deal of our DNA does not appear to code for any proteins, and I'll mention why that is a little bit later. But the take-home... Sorry?
Is there a standard for how many base pairs are needed to are in a gene? Or does it go?
It's highly variable.
Species correlated.
And you get complications in the sense that... So a gene is not necessarily contiguous. So there's one I was looking at a few days ago for a project that isn't going to happen anymore, called SERPINA1, which does something in your liver which makes your lungs work. It's interesting but irrelevant. And so the gene itself is perhaps 5,000 bases long. But it's actually split into, so you have a rotor region, a start signal, a chunk of gene, then a splicing site, it's called. And it says the gene stops here. And then there's a long stretch of what appears to be noise, just nonsense, then the gene starts again. And there's another splicing site, then there's a long stretch of noise. No, because what the RNA does is quite clever. We'll get onto that in a moment. So if we hit the next slide. So we have here, if you like, your gene. So we have a sequence here, which acts as a binding site for this huge complex of proteins to come and stick to. Exactly the makeup of these proteins varies to some extent between cell types. It's enormously different between bacteria and between eukaryotic cells. The eukaryotes are sort of plants, mammals, most big things, really. It's true. It works. It holds up as a definition. So this protein complex assembles on the binding site, recruits an RNA polymerase. Polymerase is any enzyme which makes a long stretch of subunits, including DNA or RNA. Recruits this. The polymerase reads along the DNA, and you get a piece of RNA that's complementary to the DNA, which then floats away to do its merry thing. The polymerase falls off and goes off to have another adventure somewhere. So this piece of RNA is a stretch of information that exactly mirrors what was in the DNA, give or take. So what's interesting about this is that it's not just information coding for a gene. Some stretches of RNA can be enzymatically active, in the sense that the information encoded in them, yes, you can think of it as a stretch of information, but it's also a physical object. As such, the sequence of RNA, or indeed of DNA, will cause it to fold up into interesting shapes and do interesting things. So it can fold into a specific shape that makes it a target for an enzyme that will chop pieces off it. Or indeed, there are some RNAs which can fold up into a shape which will then cut itself in half. It's fantastically complex stuff that I don't know the details of very well, but it's awesome. In every sense of the word, if you're a huge nerd. So at this stage, you have splice events happening. So if you have sort of real gene noise, real gene noise, real gene noise, through a combination of different processes, the noise will get cut out and thrown away, and you'll be left with something shorter than this that just encodes the actual information that you want. There are further complications involved in this, in the sense that you can have several results from this process. So you have one gene, which encode lots of real bits and lots of noise. The noise gets thrown away. The real bits get put together in various orders, or some but not all bits are used.
Is that a folding feature, as the way it folds? Or...
I don't know the details. Some of it's likely to be because of the secondary structure, the tertiary structure, so how it folds together. Other bits, perhaps other things, I honestly don't know. I know there are some viruses which make use of it extensively, because viruses are very limited by their size, partly because building a big virus takes a lot of energy and time, and a virus needs to minimize both of those things in order to make loads of copies of itself, or so self does. Also, simply as a physical size, as a physical object, a virus needs to be tiny to propagate well and to avoid the immune system. So viruses extensively use many genes produced from very small amounts of DNA with overlapping genes and disvariants. And so there are some viral RNAs which fold into quite complex structures. Yes, we fold like that to end up with different sequences being made from the same structured DNA. And then also some which have varying. They have they're sometimes referred to as slippery sequences. So it's a sequence which the polymerase might read through, or it might get there and fall off. For reasons which are well defined, but I have no idea what they are. There are just some sequences the enzymes aren't particularly good at dealing with. So yes, we have our DNA, structured information. Then one sequence recruits a big bundle of proteins. That bundle of proteins recruits the polymerase. So the polymerase reads from the DNA and makes RNA. And this RNA goes off, may or may not be processed, and is now, you end up with a message encoding the protein you're going to build. So if you could hit the slide. So the RNA is a template for protein manufacture. And the way it works is we only have 4 letters to play with.
What's the answer?
Sorry?
Molecular RNA.
Messenger.
Messenger.
RNA can do many different things. This is one of them. It carries a message. So obviously 4 letters doesn't give us a lot of room to play with. So what happens is we've evolved in such a way that they're read off in triplets. So now you have 4 times 4 times 4. You have plenty of information space to play with. Thank you.
Sorry.
I'm a biologist. I became a biologist because I like science, but I can't count. I don't know who this is, but I like them. Okay, they're not in EU Maniac. Nice to hear. So yes, and so they can code for this is the start of a sequence, this is the end of a sequence, and they can also code for one of 20, depending how you count it, amino acids. Amino acids are these subunits. Chemically, they're interesting, but we don't need to go into the details. So the amino acids are recruited to what's called tRNAs, transfer RNAs, but again, not terribly important. So I have no idea who that is. Yes, so the tRNAs bring them in, match them up with the RNA. Then this huge complex, which is called a ribosome, brings them together, joins them together, and so you have here a dipeptide, 2 amino acids stuck together. And the ribosome complex chugs its way along the messenger RNA, leaving behind it a protein. And on YouTube, there's some really gorgeous videos of this process in action, and I recommend you look at them, just because they're really cool. It won't help your understanding very much, but this is cool.
All the videos, they're not, I can't view the stuff, it's way too small to see.
Computer simulations. So we have extraordinarily good detail on the exact structure of these things, and so based on that, we can predict the interactions with a decent amount of accuracy. And then you give all that information to a nerdy artist, and really cool stuff comes out. Yeah, plus or minus some artistic license. Because obviously this is, the inside of a cell is a very complex place. You have a big soup of things swimming around very close together, and some are floating around randomly, as these things are, the transfer RNAs and amino acids, more or less. Although they're going to be concentrated more in some parts of cells than others. Whereas some other things are being dragged along on, they look like trains. You have these big tracks within the cell, and molecules which go along and dragging stuff behind them. Again, it's awesome stuff. Your cell is so much cooler than you think it is. So yes, so you have these peptide things, and so because each of these, again, as well as encoding information, is actually a physical object, these are what then folds up into enormously complex shapes, which we're not very good at predicting. We have some ideas, but it's computationally extremely hard. I've been assured it's NP complete. I wouldn't want to come onto that. So, and these are these fold up into enzymes or signals or fluorophores or all sorts of cool things we can play with. So these are the end result ready. And if you go on to the next slide, these are some examples. So this is one style of representation of a protein.
Richardson diagram. They not?
I honestly don't know. I know them as ribbon diagrams.
Sorry?
I know them as ribbon diagrams. I don't know.
Nobody gets them when you invented them the credit.
Yeah, okay, cool. And they give you some information about the structure of the protein. So the squiggly things are called alpha helices because the amino acids are shaped in such a way that they form into a helix. These are called beta sheets because they form sheets. These wiggly bits just aren't all that interesting, although they may do cool things. So going across, to give you an idea of the range of diversity in structures, The one right the way over there is hemagglutinin, which coats the outside of the flu virus. The top is responsible for, as I recall, interacting with red blood cells, also binding the outside of epithelial cells to afford cell entry. And when the pH changes, it dramatically deforms, exposes an enzyme site, and drags the rest of the virus out of a capsule the cell builds around it to try and destroy it, and into the cell proper so it can carry on with the infection. It's an enormously complex thing. It's absolutely beautiful the way it works. The next one along is hemoglobin, and you can see it's a tetramer. So this is actually 4 proteins stuck together, 4 identical subunits of. So there's 4 identical proteins that have come together into what's called a quaternary structure, like a 4th order structure. There's 4 proteins stuck together, and some iron complexes stuck in there as well, which is what holds your oxygen in your blood as you're going around.
What's the purpose of the 2 different colours? Is that just for clarity?
Yes. Yeah, purely so you can see which is which. I should say that hemagglutinin is in different colours. It starts off as one protein, and then during the maturation of the virus, it snaps in half, effectively. It has an enzymatic help, but it breaks in half. This is green fluorescent protein, which is from a jellyfish originally. Enzymatically very dull. It reacts with hardly anything at all. But if you shine UV light at it, it glows a brilliant green.
Put it in use for beer.
This is something I really want to do. It's like slightly illegal and potentially slightly dangerous, but it'd be awesome if it worked. If you get used to it, brewers used to express this, you'd end up with UV reactive beer. How cool would that be?
Or you can just drink G&T.
Well, it's less cool.
How dangerous?
Depends on gin.
Yeah, how dangerous would that be?
I've not found... If you can feed large amounts of it to mice, and the mice don't suffer, to my knowledge, no one's ever done a toxicity trial on humans.
What's the legislation of that?
I'll get onto that, it's complex.
Depends, will my kidneys detect it? In which case, light show.
I'm confused with security services. You all say, why are you glowing?
And this last one is called retinoblastoma. Enzymatically, it doesn't do a huge amount. It does have some activity. But this is one of the key things in the cells responsible for us not having cancer. So its main role really is governing other proteins' activities and preventing the transcription of certain genes. So it stops certain genes from being read from DNA to RNA. So you can have an enormous diversity of functions. So acting as a sort of metal chelator for carrying oxygen around the place, glowing different colours, regulating other genes, or doing really cool stuff, sort of unfolding and cutting through various substances. And so proteins really are what we talk about mostly when we're talking about sort of genetic engineering and manipulation. There is a lot more. There are all sorts of sugar structures, fat structures, and so on that you can have enormous fun with, but they're less well understood, largely because they're much harder to work with. And that's less true in the last perhaps 5 years or so. But it's rapidly growing. But proteins are best understood, and are the ones we have the most fun with at the moment. So if you go on, don't mind. So to recap that, so we have DNA, RNA, protein work. Yeah. So you can see in the nucleus, this is in a eukaryotic cell, so a cell like we all have. A bacterial cell is the flow of information is the same, but the organisation of it within the cell is somewhat different. But this holds true in bacteria and yeast and anything else you want to play with. So you have your DNA, which for everything we're going to be working with is double-stranded. The DNA gets copied into a length of RNA, which just matches the sequence perfectly, give or take mistakes by the enzyme, which happen, but rarely. This comes out, the nucleus, and the messenger. It may or may not undergo splicing into a slightly different form. And then you have your messenger RNA, so the DNA, the messenger RNA, chokes through the ribosome. The ribosome turns it into a lovely protein that folds itself, or is folded by processes within the cell, and does something cool on hopes. The rider to this is, so this is showing a typical situation inside a eukaryotic cell, so human cell, animal cell, plant cell. Excuse me, which is great. However, this sequence on its own, the peptide on its own, the sort of nascent protein on its own, will not necessarily fold itself up and do exactly what you want it to. There are very complex and very elegant systems set up within cells to take this and take it to specific places within the cell, treat it in certain ways, and modify it. So whether that means helping it fold, cutting bits of it, attaching new modifications to it, it's all fairly complex. And sort of, even though I said this is structural, the structure still encodes information that the cell can recognise for all those structures. And the problem is that the system to do this in eukaryotes, in us, is rather different to the system set up to do this in prokaryotes, in bacteria. And this raises problems sometimes. So with an awful lot of proteins, GFP for example, you can take it out of eukaryotes, jellyfish in that case, stick it in a yeast bacteria, it will be expressed absolutely beautifully, it will work perfectly. There are other proteins, none that spring to mind right now, there are many, trust me, because some of my friends are having nightmares about them in the moment, that if you stick them in bacteria, nothing much will happen. You'll get something that's almost right, but not quite there, because the bacteria simply can't do the processing you need it to. There are tricks and there are ways around that, but it's something you need to be aware of if you want to take a mammalian protein or a protein from eukaryotes and grow it up to a large number of bacteria.
Can you generally go the other way? Can you go prokaryote protein into eukaryote?
I don't know.
Or is it just switching between it?
I would expect there'd be problems, but I have no idea, to be honest. I'm not sure it's been tried.
Case depends on there have been genes that have been transferred in both directions, and depending on the type of
There's the
function.
ruby fluorescent fluffy. Ruffy.
Does that exist?
Yes.
Awesome.
Is it cute? Yes.
Although the ruby fluorescent thing is, I think it's a modification of the GFP. So it would have been a eukaryotic gene originally.
Sorry.
But yeah, I mean,
No.
okay.
Sorry.
Yeah, so that's a central dogma. DNA, RNA, protein work. It kind of works. There are some issues along the way. So if you can hit the next slide. Because we're scientists, we really hate dogma. And so we proved that wrong. So it's not wrong, but it's very incomplete. So I said DNA is an information source. It's true. But because, again, it's a physical object floating around in a complex environment, the DNA sequence it can be structural, it can be regulatory, it can be coding, or all three. So there's some stretches of DNA which code for a protein, but also fold themselves into a complex structure. And that structure is necessary. It's one of the big problems with cloning and IVF and so on. You can take the genes out of one, the genome out of one cell, stick it into another cell. But one of the problems is that, although the DNA, all the information is there, it's not necessarily folded into the right shape. It doesn't necessarily have the right modifications attached to it. So the DNA, yes, it's information, it's also a physical object, and you can't forget that. So there's some complications there. So, similarly, the RNA, it can be just a string of information. It can be structural, it can be a regulatory element, it can be enzymatically active, it can code for stuff, or it can be all or none of them. And there's enormous fields there. And there's meta information, so information about how that information needs to be treated, like transporting things around the cell. So it's potentially a very complex field. However, this always holds true. To get a protein, you need to start with RNA. To get RNA, you need to start with DNA. And for the most part, although all of this is important, for the most part, you can get away not worrying about it all that much. I put it in because anyone who's interested in biohacking may run up against it. Also, just because it's really cool. But this is good enough for most cloning work. Most of the sort of biohacking projects that, in my very brief dipping my toe into it, I've read about or encountered. So yes, bear this in mind, but for the most part, the central dogma is good enough. It's a useful line, if you like. So if you move on. I mentioned cloning. Cloning is not about making copies of whole organisms, tragically, because, my God. It's a big disappointment. So when we talk about cloning, instead we're talking about generally taking a gene out of an organism, sticking it in another organism, plus or minus modifications. It has various applications. So, for example, in some of my work, there's a protein I wanted to study. I wanted to find out where it goes in the cell. So I took the protein I was interested in, got the sequence for that on the structure of DNA, and the sequence for GFP, the glowing protein I was talking about on deep DNA, and just slap them together. And then get the enzymes to read through that, so DNA, RNA, protein, and you end up with a protein, the protein I was interested in studying, and on the end, a little tag that was a glowing protein. And suddenly down a microscope, you can see my little proteins glowing as they move around the cell.
What is the difference with that type of cloning and a chimera?
Chimera, you can talk about chimeric proteins, because that counts as, yeah. Chimera, just, as far as I know, just means two things stuck together. So you can make chimeric organisms. You can do really unpleasant things to zebrafish by taking a zebrafish blastocyst, so a bundle of cell forms before it's a fetus but after it's an egg. And you can cut them in half and stick them together and do different things like that. And they'll grow up to be organisms like that. But you can make chimeric proteins where you take half of one protein, half of another, jam them together exactly as it describes.
Okay.
Yeah.
How do you check that you don't change your results by shoving things like GFP onto it? So, I mean, how can you be sure that whatever gene it is you've been expressing with GFP stuff to track it around the cell, you don't then alter how that protein's works within the cell because it's got GFP stuck to it? Do you just have to hope and then shove it unmodified back into something else and see what happens?
You basically close your eyes and cross your fingers. It's an important question that isn't asked enough, actually. So
it's just basically you take it out and then see what happens.
It can be quite subtle, yeah. There's one called LC3, the protein I was interested in, that has GFP tagged to it, a version of it. And what you're looking for is, under certain conditions, does L33 clump together? It's a sign that something interesting is going on inside the cell. And you look down the microscope and say, yes, that's fantastic. But it turns out, and lots of people have published on this and said, yes, we've seen the clumps, glowing green, it's great. And then someone else pointed out that actually they just tend to form clumps with this tag attached. And so, yeah, it's people don't watch it.
A hit and hope kind of thing.
Kind of, yeah. GFP is normally pretty friendly, but even with GFP, since it's relatively inert, doesn't really do much, it creates some problems. And the more sort of unconventional the cloning you're doing, the more that becomes a problem, simply because we're so bad at predicting how proteins are going to fall together. I mean, you would hope that if you have one complex protein, another complex protein, and then just a string that you know is going to be a boring string of amino acids in the middle, hopefully you'll get a complex protein there, a rope, and a complex protein there will be tied together. But it doesn't always work, and it's very hard to pin down exactly what's going on. You need to be friends with a crystallographer with a lot of free time and an awful lot of money to resolve that for you. And it's kind of something that none of us can really hope to achieve. You need a university department or a research lab.
Sort of 3 kilometres long laser.
Yes. To get Sam drunk, he'll do it.
You can get electron microscopes for £2 grand.
I've got a crystallographer
I've got a couple of friends who work at Diamond. Well, I work at RAL, which has got the Diamond nuclear source next to it. I can't sneak people in, unfortunately.
I know someone who works at Diamond and they said they want to be crystallographised.
Even better.
I've talked to a crystallographer from Diamond a while ago. It's really cool. It's talking about
Diamond's brilliant. It looks like someone's landed a UFO.
Was she a blonde woman called Anna?
No, it was a blonde bloke whose name I can't remember.
Not Anna.
Aren't there a lot of crystallographers at any given synchrotron?
What's that, sorry?
Aren't there a lot of crystallographers at any given synchrotron?
Occasionally on my knees begging, but yes.
Yeah, but blonde is a subset.
I don't know, a lot of them tend to be from Sweden.
As a running gag, actually, all male crystallographers have beards. And it seems to be true. And the idea is that you need a nucleus for any crystal, right? So you lean over the bench and you're scratching a beard, and suddenly crystals form. And I've never met a male crystallographer who doesn't have a beard. It's freaky.
At least a worrying question of what about the female crystallographers? What are they scratching to form nucleus cells?
I've seen this done like this.
They have beards too.
Yeah, anyway, cloning. So, yeah, next slide. So the aim of cloning is to end up with a plasmid that we can put into our target organism where it efficiently extracts our desired gene. A plasmid is just a circle of DNA that has a replication origin on it and your gene, and typically some means of selecting the plasmid. So a replication origin, I mean, when the cell divides, the enzymes responsible for reproducing the cellular DNA, so that both daughter cells have the right amount of DNA, will also latch on to the replication origin of the plasmid. So as well as making a copy of your DNA, or the cell's DNA, they'll make a copy of the plasmid's DNA. And so each daughter cell will both have a complete set of its own DNA. It will also have a complete set of plasmid. You get various numbers of plasmids per cell. And it's really interesting and a very complex thing going on in bacteria, because a lot of bacteria have many plasmids performing essential roles. Ask me some other time. It's great, but I'm already running over time. And the use of these things kind of includes protein production. So you have a protein you want to make in large quantities, stick it in some bacteria, grow it up. improve survival through antibiotic resistance, reduced survival, so you can give bugs new vulnerability to drugs if you particularly want to. You can modify their behaviour, so new reactions to stimuli, so the way they swim to water away from food and nutrients and so on, and for creating fusion proteins. And your imagination is the limit to some extent. Yes, please. So as I say, the plasmid circles today you can put into many cell types. As far as I know, you can put it into more or less any cell if you design your plasmid directly. And use them to carry a gene of interest and target an organism. Now, the tools we use for cutting up the DNA, called restriction enzymes, these are enzymes, so they're complex proteins, that they recognize specific short DNA sequences, bind to that DNA, and then chop it. And what they do is they leave sticky ends, which is overhangs which allow the DNA fragments. So this is EcoR1, which is a very cheap and widely used enzyme. It recognizes this sequence. So any DNA sequence that has this floating around in the tube, you throw in EcoR1 to the same tube, it will bind to the sequence and chop it here and here. And so you're left with a sticky end. And so you can separate these out. And now if you put this fragment and another fragment with sort of a matching end into a tube along with another enzyme called a ligase, this and this will line up, and then an enzyme called a ligase will come and glue these together. So what we're able to do is cut a plasmid, separate out. So this example here, this is actually something I did a couple of months ago. So there's an enzyme site. There are loads and loads of enzyme sites all the way around the plasmid, but I've just shown the ones I'm interested in. So I wanted to get this gene into this plasmid, where part of some work I was doing. So I could cut it here and here with different enzymes, and then you can separate out using electrophoresis, which I'll talk about in a little bit, the big fragment and the little fragment that we actually want. Simultaneously, we cut here, in this case with one enzyme, or you can do it with two different enzymes if you want to drop something out. And then we take this fragment and this backbone, put them in the same tube with the DNA ligase, and then the sticky ends of this overlap with the sticky ends left here, and it joins together to form a full circle. And we can put that inside a bacteria and grow them up and get huge amounts of the plasmid out, which will then go on to use in the rest of my project. I feel I haven't explained this very well. Is that okay?
I think it's fairly straightforward, but I do wonder how much experimentation there is in finding the right points to cut off the sequence, the right enzyme to find the right enzyme that can actually cut at that position.
It can be tricky.
To find segments that can actually fit together. So there are loads of open questions, and then in the end you might not even know what the outcome will be.
Yeah, it can be tricky. So basically, you need to know the sequence of your whole plasmid, and then if you know the sequence of your plasmid, just a string of letters, and you know what sites all of your enzymes, your library of enzymes will recognise, you then just get a computer program to go around the map and label all the different enzyme sites, and then you spend the best part of an afternoon trying to find sites that will cut near your gene and will also cut where you want to put it.
So is there something where software helps you, or where you actually need to experiment? Or where you have databases to look up stuff?
There are tools to do this, so you can tell. You can tell.
Sorry, match the patterns. Is that right? There's some kind of regex is going on that you can match the patterns on.
I presume so. Yeah. So there is software you can say, I want to cut this gene and put it here, and tell it various tolerances, and it will say, I recommend you cut with this enzyme and this enzyme, and then this enzyme and this enzyme, and put them together. So yeah, it's computationally quite trivial to do. But the software I have on my work computer isn't all that good. So
does that mean you get leftovers that you weren't expecting whenever you're kind of this kind of thing? Because you'll get sort of artifacts between the two joints.
Yes.
So how often does that actually work, and how often does it go horribly wrong and explode in your face?
Generally, it works pretty well. If you really need very precise, cuts and very precise fragments, there's a way around it, which I'll talk about in 2 slides.
Is there a minimum and maximum size for your software?
Yes. I couldn't tell you what it is offhand. I happen to know this one is. I happen to know. This one's about 4,000 bases. That's about 8,000. It's somewhere around there. If you get too big, the bacteria can't really deal with it anymore. So upwards of about 9 or 10,000, I would guess. And the lower limit is, I don't know, how small can you make a circle?
I mean, as much as you can do it with like 20 base pair, but then you cannot carry any information. It's just.
Okay, so that's cloning. So if you hit the next slide, there's perhaps a better picture of it here. So you have your vector, which you cut, and your fragment, your insert, which you again cut. Put them together, and some enzymatic magic happens, put something else in the tube, and they get joined up. And you put them inside some bacteria. And when this bacterium divides, you have 2 daughter cells, complete with all of the bacterial DNA, and hopefully all of your plasmid DNA.
So which one does it choose to express if you've got? Are they sort of mutually exclusive in what they tell the cell to do, or
they'll both be expressed.
Okay.
Yeah.
But there must be conflicts, presumably, or is it designed
Not
to
too much.
use conflict?
No, so this will have a site in it that this will have the same site in it that the bacterial DNA has in it, in order for the DNA polymerases to bind to it and to copy the DNA. It will quite happily go for both of them, pretty much indiscriminately. You do run up against some issues because it costs energy to make this. A bacteria without plasmids will generally reproduce faster than a bacterium with plasmids, just because it's hard work to make them. To get around this, as well as your insert, you normally put an antibiotic resistance gene in, and then grow the bacteria in medium that contains antibiotics. So anything without this dies because of the antibiotics. Anything with this can survive and reproduce. And so you introduce that selective pressure in favour of your inserted gene. But they can coexist quite happily, and most bacteria will have, or at least many species of bacteria, will have lots and lots of plasmids floating around doing various things of varying importance in bacterial survival. So indeed, and antibiotic-resistant bacteria tend to have the resistance on plasmids that gets. So rather than being spread purely through hereditary to the daughter cells, the plasmid can actually come out of this bacteria into its neighbour. So almost between sort of sisters rather than mother to daughter. And that's part of the reason bacterial resistance spreads so fast. But yeah, there's no problem with having them both in the same. It's a pretty natural thing. One of the useful restriction enzymes is once you've tried to make your plasmid, you're just looking at a tube with liquid in it, who knows. You can then take your new plasmid and your new map that you put together, because you know the sequences you put together, and then cut them up. Why am I doing this? I have a laser pointer. This is awesome. Oh, yes.
A laser pointer.
And then cut them up with restriction enzymes, and you know that on this map I will be able to cut here and here. I expect one fragment, 8,000 bases, one fragment, 1,000 bases. And so this is a gel I did sometime back in February. I can't really remember what it's for. Checking human factor 7 long poly A, no one cares. Digest was an enzyme called HEAT2 for 4 hours. And so these have given me what I was expecting. These three here. This one, no clue what happened there. Something wrong anyway. These two look alright. That's wrong. And this lane here is supposed to be empty. This one, not a clue what happened. Something went wrong as well. So, as well as deliberately cutting these things up in order to do cloning, we can use it as an error checking to check how plasmid is doing what we think it should be. Again, all making sense? Yeah?
What is electrophoresis? It's a thing.
Wait, like 2 minutes.
Okay.
So, next slide, please. Alright, so I've already said this. So you have your bacteria which are antibiotic resistant, and some of them won't have taken up plasmid, so they won't be antibiotic resistant. You just grow them in medium with antibiotics, plate them out. So with no antibiotics, you get loads of them, and only one which you're interested in. With antibiotics, the only colonies you get coming out are the ones you like. So the end result, hopefully, is a pure culture of beasties which are expressing the novel combination of genes you've chosen and the conditions you've designed. You hope. It may or may not work, and of course there are going to be intermediate steps. You may need to do several rounds of cloning to slowly assemble the plasmid over several generations. It can be quite tedious and frustrating. It'll either work first time perfectly, or you'll have a nightmare trying to work out why this particular sequence doesn't want to be cloned. Because, as I said, there are some sequences that will fold themselves into weird shapes, or they have weird chemical issues going on with them, and the enzymes don't like it. And there's nothing for that but trial and error, unfortunately. There are some known tips and techniques. The most popular one is giving it to a PhD student and said, "Do this." Apparently tears work as a reagent. So, next slide. I forgot what this is. Okay, so some of the core techniques we're using. So, perhaps one of the most important techniques in modern molecular biology is the polymerase chain reaction, PCR. It's a technique for taking a stretch of DNA and making an enormous number of copies of it in a very short time. And it was revolutionary and invented repeatedly. Can you remember the guy's name? I'm really bad at names. Anyone?
I've never heard of it.
Sorry? I believe you. Okay, but the way he tells it, the idea came to him whilst he was driving way into the small hours of the night along a highway, in America, smashed on acid. And that moment was the inspiration for all of modern molecular biology. So do drugs.
Carrie Mullis?
Yes.
Yeah, Wikipedia says so. Right.
So what happens anyway? You have your double-stranded DNA here. You heat it up very hot, 95 degrees or thereabouts. It splits into two because they just fall apart at that temperature. You have a primer, which is perhaps 20 bases of DNA that matches the end of the sequence, and at the other end as well. And DNA has a directionality. So this primer acts to start a polymerase reaction, and it will go this way. This primer also has to start a polymerase reaction, and it will go that way. So you attach your primers here by varying the temperature, and then your polymerase, which is also in the mix, starts working, and makes each of these into a new double-stranded piece of DNA. So you had one copy, you now have two pieces of double-stranded DNA. You go through this sequence again, each of these gets reduced into two. You go through this again, each of these gets split into two, and so you have an exponential increase in the copies of DNA up to limits imposed by how much reagents you have and sort of what's going on to you, basically. And so within 40 cycles, you can get colossal quantities of DNA out at a very, very low number. And it can be used for a lot of things. One of the applications is cloning when you have very precise requirements. So you can put your plasmid of choice in and design one of your primers. So the first 20 bases of it or so will match the edge of what you want to clone. And then the 5 or 10 bases behind that will be a restriction enzyme site, which you'll then use to put it into your new plasmid. So you'll end up with loads of double-stranded DNA that has your fragment of choice, and on the end of it, there will be a site which you can cut with a restriction enzyme and then drop it into the plasmid that you're trying to put it. And it's slightly more work than straight cloning, but it works beautifully. Getting hold of these primers is very simple. Depending on the order it from, it's 20 pence per base and then post it to you. It is correct.
How long does each cycle take, sorry?
Depends on the length of your fragment.
Okay.
So the ones, as a ballpark, the ones I typically do is, the cycle I normally do is heat to 95 for 10 minutes initially. It's magic, it works. And then 95 for 30 seconds, 60-ish for 30 seconds, 60-ish for 30 seconds.
Is 1,000, the polymerase is 1,000 nucleotides per second.
Sounds about right.
Yeah.
So was that sorry?
The enzyme with the reaction with polymerase is what can attach 1,000 nucleotides per second. So this, how to say, this implies diffusion.
Yeah. Usually times per second.
Yeah. So it's very fast.
Question: are those temperatures and durations in temperatures in Celsius?
Yes.
I thought that proteins are denatured around about 40-45 degrees.
Some proteins. So yeah, that was a huge limitation when this was initially done.
It's not protein.
Yes, but the polymerase in the mixture.
Yeah, but the polymerase is one. Yeah, sorry, it's one of.
So when the technique was originally done, you had to after every time you did this to separate the DNA, you had to add more polymerase because it was destroyed. But what we use now is called Taq polymerase, and it originally, there are many different ones, but the original one came from what's the word? Heat-loving. Thank you. Yeah, thermophilic bacteria, and it's perfectly happy at 95 degrees. It's since been modified to make it more robust and more stable. And there are now several different enzymes from several different sources, but they're all fine up to 95, 100 degrees. So the variants in the PCR. So you can do what's called reverse transcriptase PCR. So you remember transcription is going from DNA to RNA. So reverse transcription. Thanks for that. RNA to DNA. So you can detect not how many copies of the DNA there are in the cell, but how many copies there are of the message in the cell, or indeed if the message is there at all. So you can check to see if your gene's being expressed at that level, or you can say under these conditions by genes being expressed more or less. Quantitative PCR. So rather than saying oh there's loads there, there's not much there, you can actually work out an exact number.
Give or take, what's the error level there?
It depends how many you're measuring. So I normally go for. Okay, so the one I normally do, we're measuring something in the region of 10 to the 12 copies in our sample, and the error region there is it's normally pretty good. It's 50% or so. So within a power of within a power of 10, though. And so, but you can get. There are people who seem to do this very, very precisely, and they're measuring they're expecting to find in the order of 10 or 20 copies in a cell, and they get very consistent results. There's nothing I've tried myself. It seems to work. So it depends on your experimental setup. In my case, I'm many orders of magnitude above that, and I don't really care about it all that much. If I know I'm within a power of 10, that's fine. And there's also mutagenic PCR. So there are techniques you can do. So polymerases that don't work all that well, which will randomly introduce mutations. And so you can start with a gene, use this polymerase, randomly introduce mutations all the way along it. So each daughter strand will be slightly different. Express these in a library of bacteria. So each bacterium, you can work it out. So each bacterium more or less gets one plasmid, and it's expressing a slightly different version of the gene. And then you can do massive screening things. So each bacterium will be expressing a protein that's a slightly different colour, or each bacterium, or you might be trying to find one that only works at a certain temperature. And so you get tens of thousands of bacteria spread out on plates, subjecting to different conditions. And the ones that change colour, or the ones that survive, or the ones that smell of fruit, or whatever, are the ones you pick and carry on working with. And so it's a very nice way of getting lots and lots of variants on the gene very quickly, instead of spending hours or weeks or years of your life trying to rationally design something, which is possible to an extent, but enormously hard, depending what you're trying to do. There's a guy in my lab, actually, he spent an enormous amount of time trying to rationally design a change for a protein to change the colour it expresses, trying to push into different wavelengths. And he got very close, but in the end, he had fantastic success just by basically doing this by sheer fluke. And then he sequenced it and found out that he was one position off. Within the next month or so, before he'd given up, he would have got it. But basically, the message here is that evolution works better than intelligent design. Yes, next slide. Western blotting and ELISA. So I've talked about so far about detecting and playing with DNA. So this, NXTs, is an antibody. We have countless thousands, millions of these floating around in our bodies. This bit is invariant. The purple bit is generated, a biologist says random, a more numbers-oriented person might say chaotic method. So each one of us has a unique library of these. They're made from DNA, but the process of making them is so complex that mine would be nothing like yours. And basically, the only selection criteria is, but it's complex. And the main selection criteria is that it doesn't bind to anything we already have in the body. And so these are what's responsible for binding to recognizing non-host, non-self. So recognizing viruses and bacteria and so on. And we can tailor these to an extent. So you can, for your protein of choice, we can manufacture, give or take, some success. Some of them work very well, some of them don't work at all, most of them are in the middle. These proteins these complex proteins that will bind to your target you're interested in. And so when this happens, we can attach a tag here and use them to immobilize or detect a protein that you're interested in. Making these is enormously difficult and involves animal work. It's hugely illegal to do at home, as you might imagine. So this is probably out of our league making them, but buying them and ordering them, I don't see why not. They're relatively expensive though. Depending what you're after, you can expect to pay somewhere from 100 to 300 pounds per tiny tube. It's a bit of a crapshoot as well. It may or may not work. But yes, next bit. And this is an example of a Western blot. So I've got lots of protein samples. I've run them by electrophoresis, which I think is the next slide. So they separate out by size and or charge, depending how you set up your system. And I put on an antibody with a label, and you can see that my protein of interest, I've diluted it down here to know it's the right thing I'm supposed to see. And these are some samples I was testing. And so this is what you get. So I know this is my protein, and you get nice thick bands saying, yes, the protein is here in these two samples, and this is band in my control as well, and not in my negative control, which is fine. So that's one of the things you use for. ELISA is effectively the same thing, you're using antibodies to detect protein's presence, but it's a different format. So you get a numerical output, which is nice, but you don't get the information to check that it's actually the right size charge ratio. So next slide. This is very information heavy, I'm aware of. I'll make the slides available for anyone who wants to take them away and read through their own time. Yes, so obtaining and checking DNA. Getting hold of these genes, relatively easy, actually. So there are many thousands of annotated gene sequences available online. A good start is the NCBI, which I forget what it stands for. Not terribly important, but it's a huge body run by some arm of the American government. There are many other databases as well, it's just the one I'm most familiar with. And it just has the enormous gene sequence. If you want, you can go on and download the entire sequence of smallpox. And then you can send off DNA sequences to various companies, and they'll send you back a gene with the DNA in it. It's...
And the company said $20,000.
Yes.
Is there not protection against ordering smallpox?
Yes.
Probably, yeah.
This is when it comes to bio experiments, this is one of the big open questions where currently there's no good final answer. How do we...
We're running low on vaccine studies. That doesn't exist anymore. Smallpox.
Well, the question is, now that we have tools to easily build these organisms, who can regulate that, and who can regulate that effectively, the use of those tools? Because in the end, a service provider that prints them for you would have to check, is that something harmful that doesn't print? But how would they check that? So how would they check that? What do they reference to identify sequences that create potentially harmful organisms?
And that's computability.
But it's also an information problem.
That's going to be a nice end.
I mean, it's a similar thing where it's like you could get all separate sections assembled by different companies and have no knowledge of the final product, assembled yourself in much the same way. You can get a gun printed by all different companies on the internet.
Yes, which means at the moment it looks very much like as soon as machines get cheap enough so everybody can buy one to synthesize organisms, it's going to be virtually impossible to regulate.
I suppose we just have to hope that there's enough people bored and sitting out there generating smallpox vaccines.
Yeah.
That's you can target regions and separate the entire code. It's just specific region you can actually it from. You can have specific targets.
That's right.
But getting away from Russia.
Yes, we are.
Well, no, I don't think we're getting away, because in the end, this conversation is among a group of people who are potentially interested in actually playing around with that stuff. And at some point you're going to expose to these kinds of questions. Maybe not this year, but in 10 years, you're potentially going to be subject to regulation or to convention. Your tools might be limited compared to what you have now. And I think this is pretty much the conversation to have right now.
Yeah, I know some people in my lab are very worried about this sort of thing. I mean, the thing with smallpox has been done. 2 or 3 years ago, an American lab, I forget which institution it was, but did this. They ordered parts of smallpox. They downloaded the smallpox sequence, ordered parts of it from a variety of companies around the US, and assembled it and made some. And it worked. And they then very carefully destroyed it.
Did they actually test it properly?
Fine, properly. I shouldn't think so.
Human subjects.
The World Health Organization claims there are 8 complete copies of the smallpox virus around the world. Two are held by WHO. One's held by the US, one's held by the UK, various other. Russia had one or two copies. Well, I say Russia, the USSR had one or two copies. When it collapsed, they were in some biolab somewhere in Russia. No one knows where, and no one knows what happened to these copies of the virus. And everyone's hoping that somewhere in somewhere in the frozen tracks of Russia, it's died. But for all we know, somewhere in Russia, someone's simply going.
In Soviet Russia, smallpox finds you.
Yeah, pretty much.
Yeah. But yeah, it's certainly a debate because it's one of those things that's going to be very hard to police. Because there's another process called. Sorry?
It's a harmful code problem. Anyone can put together lines of code, and you can sit there and get bored and write an algorithm to write code for you.
And if you're feeling particularly tricky, there are techniques you can use. So you remember I was talking about this, the number of letters you have in the DNA codon, the DNA triplets, 64, the physicists shouted out. There are 20 amino acids you can code for. Now there's some redundancy in there, so there are things where you have 3 or 4 different codons that encode one amino acid. And so you can take the smallpox virus, rewrite it completely, so the DNA sequence will look nothing like the original. But because of this redundancy in the codon design, it will still encode for the smallpox proteins. Now, how well it would actually work is a different question because changing the DNA sequence is going to change how effectively the enzymatic reactions work upon it. But it's another huge problem. So you can't just scan sequences and say, does this match up to something nasty?
So how many times can it be mutated from, like, how many times can it be transliterated from one to the other? I mean, are there, like, 10 different things it could become? Or, so,
Well, if you imagine, I don't know how long smallpox is. If we say arbitrarily, smallpox is, like, say, I don't know, 3,000 bases long. So that's 1,000 codons. For any given codon, you can replace it with, it's an average of like 2.5 others. So some of them there are 4 triplets that encode for the same amino acid, and some there's only 1. It's something like 1,000, and so it's 1,000. Yeah. I mean, again, it's something you can do computationally just by iterating through every possible combination. How hard that is, I can guess. But certainly the sheer volume of orders.
Many.
Sorry?
Yeah, I was going to say 1,000 choose 2.5. You're talking factorials of 1,000.
Yeah.
Which breaks most computers.
And given the sheer volume of orders going into these companies on any given day, I mean, I order PCR primers most weeks. Loads of other labs order much longer sequences. It's a tough problem, and it's going to be a big one.
And additionally, as you say, it's a public service. Everybody can potentially
absolutely, yeah,
have sequences printed.
I said, there's this debate. I mean, do you need a big license to order DNA? Currently, no. Will you need to do it?
So you're saying I should do it now before I license it, right?
Yes.
Stock up on your bio warfare now, yeah.
In 5 years, you heard it here first.
There are some technologies coming down the pipe which will put it, not so much in the region, but into like, I don't know, sort of £5,000 or £10,000 for a box that will sit on your bench. There are sort of, yeah, and there are labs working all around the world on this kind of thing. It's going to come, and it's going to come soon. It's a problem that we need to worry about.
It's actually it's a problem in terms of the technical evolution. It's very comparable to computing development, because at the moment there are limitations in the lengths of sequences you can print, which is something that's constantly improving. And obviously there are going to be different processes and different drawbacks and so on.
The reassuring thing is that most attempts to make new pathogens don't work because we are very, very good. Our bodies are phenomenally good at combating pathogens.
Go on.
Yeah. So a round of applause for ourselves. We're great. And pathogens have obviously been evolving with us for however many millions of years, depending on pick a pathogen for the time span. And so they are extraordinarily finely tuned machines. We don't understand fully, we have no clue effectively how we work, how we battle with pathogens. We have an astonishing amount of data, and we know that lots of it is very elegant and very powerful, but we know enough to understand that there's a hell of a lot more we have no clue what's going on with it. And similarly, we don't really understand what the virus is doing. So you take a virus which we don't fully understand, ourselves we have no idea what's going on. It's enormously difficult to sit down and design something that's going to work efficiently in that space.
So the summary of this is, but when we get around to printing our own viruses, we're stuffed because we will wipe ourselves out by just not knowing what everything does.
Well, no, I think if anything else, it's the opposite. We'll fail to make anything that does anything interesting. The one that kills us will be an accident, I'm sure. But
script kiddies, I can just see.
And the one thing there is expertise on is building vaccines and
I mean,
maybe because it's more interesting to research that than things that are going to kill us.
So,
It's certainly
once again, I'm thinking script kiddies. A genetic algorithm to print out every virus possible.
That'd be really rich script kids.
Yeah, but you know they're out there.
Yeah, it's true.
They've got your credit card to pay for.
Good luck to them, there's no money on it.
Yeah, about that way. Yeah, so it's a big, important debate that is going on around the world at various levels. And it's worth having. But can we hold it for like 20 minutes? It'll be less time now, I'm almost done, I swear. So, next up, please. Yeah. Right, so setting up a lab. So we're talking about some of the techniques. So obviously you need the right environment, friendly, supportive, cuddly. But it needs to be cleanish. You need containment and disposal. Containment is a biggie. There are quite strict laws governing how we dispose of genetically modified organisms in the lab. My understanding is that some biohacking communities are getting around this by working on organisms that are fundamentally a little bit wussy. So they'll choose an algae or a bacterium or something that simply cannot survive in this environment. Either because it needs massive salt concentration to survive, or a particular temperature, or a weird nutrient, or something like that. But containment's an issue we have to consider. Because, as I said, creating a plague is very unlikely, but it is a bad
You
enough
wouldn't want that
consequence.
on your conscience.
Yeah,
would you?
and disposal as well, because while a lot of work is going on to minimise the use of harmful chemicals, there are bound to be some, and the council get weirdly picky about what you pour down your drains that melts through them. It's just picking hairs, I know. Reagent handling. So we need to be able to measure tiny volumes and masses, and tiny, so in the order of microliters. So a teaspoon is 5 mils. You need to be able. I think a sensible minimum to aim for is 1,000th of that.
But speak to somebody stupid. What's a reagent?
Ingredient.
I.
Active agent. And in terms of masses, sort of tenths of a gram, it's actually can be quite difficult to buy these balances because the only people who buy them are running labs. The only people running labs are either in universities or businesses, or they're making drugs. And so, if you start buying these kinds of equipment, at least through reputable sources, or disreputable sources that police are watching, they will turn up and ask questions. And it's all about how charismatic you are and say, no, it's fine, I'm running a lab, but not to make drugs. It's not illegal to have them. To be clear, it's not illegal to have them, but you will raise eyebrows if you get noticed. Centrifuge is pretty important. So basically, you need to be able to separate stuff out depending on mass. So pelleting, if you have a culture of cells, you give them a spin, all the cells go out to the bottom, you can pull the culture off. Or indeed, you can save the culture if the cells are releasing something into it that you want to keep. And if you want to work with pure DNA, you need to be able to spin it fast enough that the DNA will pellet at the bottom, and the rest, and you can pull the rest off. And actually, Johnson's this thing. This frightened me quite a lot. The Dremel-fuge. So you can clip tubes into this, stick it on a Dremel drill, and spin it.
Don't do
it. And what will happen is it will get up to about the right speed. So rough calculations I'm doing based on this, we can easily hit sort of 10, 11,000 G at the outside of the tube on this, which is enough to pellet DNA. And then it'll ping off, ricochet off the ceiling, the wall, the floor, another ceiling, miss me, and then land.
In someone else's eye.
Yeah. So this needs refinement, but it's not a huge problem as long as you get someone else to hold it.
There are much better ones. That one was a crappy print that we did, which hasn't come out very well. If you go and order one from one of the many online printing sellers, you can get a metal one made, which is way better and doesn't ping tubes off in your face. So working with bigger volumes is also advantageous. So tomorrow morning I'm going to be doing the same thing,
So spinning things at 6,000 G and then later 18,000 G, but there's going to be like 6 half-litre volumes. And if one of those is not perfectly made, the centrifuge, it'll wobble, then it'll go bang, then the rotor will execute the side of the centrifuge, through one wall, through another wall, then rebound off the last wall. And this happened in my undergrad before I went there. It's in some institutions it's an urban legend, but it has happened in the past. I know people have witnessed it firsthand. So that's now kind of funny. Yeah, so there's these things. Big centrifuges can be terrifying, and they're very expensive. So we need to find ways to avoid using them, basically, or just be extraordinarily patient and just sit there and watch it settle slowly over the course of 4 or 5 years. We need a thermal cycler to do the PCR. They can be expensive. There are projects going on for open source. We need an electrophoresis tank.
Hold on, what is that?
Thermal cycler? It's basically a programmable hot plate. So it'll heat the samples to 95, then take them down to 60, and then back to 70. Yeah. Electrophoresis tank, which I'll talk about very soon, coming any moment now. I just think this will not set the sky. An incubator and a water bath. You need to keep them water warm. It's easy enough. So incubator to keep your bugs half growing whatever temperature they like. Water bath, most enzymes like being 37 degrees. So you just dunk them in a water bath that's at 37 degrees inside a tube. And let the reactions carry on.
And is that the same regardless of organisms? I know the human body temperature is 37 degrees. You get the same across other life forms as well.
Yeah. It varies quite a lot. Most of the ones you buy commercially will be optimized to be around 37ish. So there is restriction enzymes, for example, they come in a huge book, or a CD. Huge books like my satisfying film, and it will tell you the temperature at once. Most of them will say 37, some want 50, some want 60, some want room temperature. So yeah, for pets, liquid handling. This is one of the most famous models of pet called Gilson. So you can see the pet itself, elegantly modelled there, don't you think? And the disposable tip on the bottom. The tips are very, very cheap, easy to get hold of. The pipettes are somewhere in the region of 100 to 200 pounds new, much cheaper on eBay. The calibration is not terribly difficult to do. So next. Great. Centrifuge. This is the grown-up version of this. So this will spin 1.5 mil tubes, 24 of them, up to, this one will go up to 13,000 G for X amount of time. As I said, it's nice to get larger masses if you can. That's a significant problem, I think, if you're working with bacterial cultures dealing with big masses.
Is there a possibility of combining longer spin times with lower Gs?
Yes, that works for most things.
Wait, why are larger masses desirable?
Just because if you're working with bacterial cultures, if you want a significant amount of bacteria, so you'll get maybe 50 cubic millimeters out of a 1.5 mil G, whereas you might want several grams, like several cubic centimeters. So just a sheer quantity issue.
Half a litre at 11,000 G.
You press it and it spins up, and it carries on spinning up, and it carries on spinning up, and you want to run, but you have to stand near it with your hand on the emergency stop button just in case.
Yeah, because you'll be able to get it in time.
I think the idea is you die, but no one else does.
Shouldn't it be a deadman handle,
Yeah. So electrophoresis at last. So the electrophoresis tank, all it is tank. It's a bath filled with a buffer with various electrolytes in it, which are quite easy and cheap to get hold of. Nothing particularly nasty in there. You put about 100, 150 volts across it. 150 is maybe too much. You'll melt the gel a little. And you'll get it to a current, I don't know, a low current. I can't remember what it is offhand in hours, but it's very low. I'll have to check. 400 milliamps or something is the maximum ours will go up to, and we never hit that. And what happens is, because DNA is charged, it has a negative charge, it will want to flow towards the positive electrode. So you have an agar gel, which is just lots of long string molecules put together, and so you get a solid object with tiny pores through it. And the DNA will try to migrate towards the positive end. It'll be dragged through, but the matrix can impede its process. And so the small pieces of DNA will go through relatively easily. the big piece of DNA will go through a lot of difficulty to can't get through the pores. And so, as it flows, the DNA separates out by size. And that's how you can separate out your DNA fragments.
How do you make sure the agar has the right size, or how do you make sure the pores are the right size?
You control the percentage of agar you have in your gel. So
it's just the fact that at the top it's going to be denser than at the bottom.
Yeah. Yes. So, yeah, and you can use the same principle for a different setup, but the same principle for separating out proteins. Ideally purely by size, but charge sometimes plays a role in it. And for RNA as well, you can do the same thing. So that's electrophoresis. It's not terribly difficult, but yeah. And then incubator water bath. You know, how hard can it be? So you have a heating element, a water pump to circulate the water, a thermostat. I never found out what this button does. I pressed it, nothing happened. I was really annoyed. And like a little float, so your tubes can merrily bob around. And it's great. The company that makes these gives them out all different shapes. I've got one in the shape of a flying pig. It's just my lucky float. It's great. And loads of crap around the place. So yes, you can float your tubes around in the nice warm water. It's useful for defrosting stuff as well. It sounds really trivial, and it is. It's presumably quite easy to make, but it's one of these essential things we need to get. So next, thermal cycler. So this is something that I don't know a huge amount about. This model. They all look very different. As a programmable hot plate, typical program, heats up for 95 degrees for 10 minutes, and then 40 times loop through 95 degrees for a minute, 54.5 for 1 minute, 60 for 2 minutes. For example, most of the programs I run are faster than that. 60 degrees for 10 minutes to wrap it all up, and then end. We normally take it down to 4 degrees because we're lazy. We just want to pick it up a few hours after the thing's actually finished. But really, an operating range of 50 to 90 degrees, 50 to 95 degrees is all you need. The big PCR machines you get are phenomenally expensive, but there are ways around that, which I guess I was going to talk about.
Any particular temperature, I mean, is that sort of 95 degrees at a full 1 minute, or is that heat until you get to 95 degrees, take the time difference from when you were last at temperature, then go, I mean, is it?
It depends on the algorithm your cycler is using. Different companies' cyclers tend to use slightly different algorithms.
Doesn't that give different results then?
Yeah, so well, not. It depends. If you're doing quantitative stuff where it really matters, a lot of them will have settings where you can change the algorithm. So you can say a really popular one, because I get the impression that one of the first big models was the ABI 7700 or something like that, ABI, I guess, because several machines I've used, made by different companies, have given you the option to say emulate this machine. Pretend that it'll rejig its algorithm, so it gives us the results of that machine if that's the one you do this stuff and set it on. For if you just want to say is DNA present or not, or if you just want to say, if you just literally want to make loads of DNA to use something else, it doesn't matter that much. There are some PCRs that won't work very well, and you have to play with various parameters, one of which is the program, and for that case it would probably matter from machine to machine. But for the most part, it's not terribly important.
What's like the minimum accuracy you can do? So if you say, I want it to be at 90 for 30 seconds, 15 seconds.
I don't know.
Is it like
you can program it to do it for 1 second.
But
what it actually does, I've no idea.
It doesn't tell you that.
No. There is a temperature probe built into some machines. I never played with it. So, I probably need to explore that. I feel sure someone somewhere will have all this data. So it needs to be fairly accurate. So I've had some reactions in which pushing the temperature of one of the stages up by 0.5 of a degree makes a difference when working or not. So for the most part it's fine, but 0.5 you really want to be able to do that. Otherwise eventually you're going to come up against a brick wall, I think. Possibly there are other sort of tricks and bits and pieces you can fiddle with to get around it. But it does make life much easier the more accurate this becomes. So yeah, spectrophotometer, we probably wouldn't need one. It's useful for some applications. Basically, it's an accurate way of measuring exactly what colour something is. So how much light of a particular wavelength gets absorbed by your sample. So in this case, it's hard to make out. There's a plate here with 96 wells in it, and they're all kind of orangey. So I want to know exactly how orange the plate is. because it gives an indication of how far a reaction in each file has progressed, which is telling me something about how much protein is in each file. Also, there are things like if you want to do if you want to make a protein that changes colour in a certain pH, which some do, then you can measure that precisely. Or if you want to measure if you try to change the colour of a protein, spectrophotometry is useful for that. It has surprisingly wide application, but most labs get by without them. But it might be fun to build.
I was going to say, it should be something that's quite easy.
Part of why I put it there. You need very accurate colour filters and very accurate light sources. This laser is actually from something similar. It's quite weak, and it's very old, so it's bigger than it needs to be. But it's also a remarkably narrowly defined wavelength that it throws out. Its original function was related to this, so you need very clean light sources and very clean detectors. But it's something that might be fun. I can imagine people having fun building. And I think that's my last. Hello. Ready? So obviously you can need some reagents, some ingredients for all your experiments. An awful lot of molecular bodies in the lab. I was really disappointed. It's none of this Max Islander stuff. It all comes in kits. I was like, put sort of 2 mils of the yellow stuff into the blue stuff. It's really anticlimactic. The kits can be fairly expensive though. And there are things like DNA purification, which you can do just with raw chemicals. But the chemicals are really nasty if you want to do it properly. Or you can do it with a kit. The kit's fairly expensive. My instinct would be always go with a kit. But you can sort of have a middle ground. So the kit is includes a tube with some resin in it, and it's set up such that the resin will bind DNA or not, depending on the industry. And it comes with buffers to give those conditions. So what you can do is buy the tubes with resin in it, but not buy the buffers, and make your own buffers. Save money that way. And then you avoid the particularly nasty chemicals by doing that. So there's middle ground to be found, and there's a lot of work being put into finding a way to make these systems less nasty and less expensive in the sort of home scene. And careful planning of your cloning, for example, can reduce the library of restriction enzymes you have. So in my lab we have, I don't know, maybe 100, 150 enzymes, each one costing between £200 and £300 for a tube. Yeah. If you're careful, and if you want to do PCR-based cloning, you can get away with maybe 2. Which, it means your work will go a lot slower, but it also means you don't have to mortgage your house and your neighbour's house. Yeah. So with careful planning and some ingenuity, you can really cut down on the stuff you actually need, I think. There will be some things going to be hard to get, including the restriction enzymes. There's a guy, Katal, who is looking into these things, and he said one of the biggest problems to run up against is restriction enzyme availability. So a few of the big suppliers are very reluctant to sell to individuals. I don't understand why. But he said he's got hold of some remarkably dangerous chemicals, but for some reason not the enzymes. It's weird. And also the community needs to settle on model organisms to work with. Versatility and safety concerns. So we tend to work with E. coli, which are sort of enteric bacteria, and the ones we use are pretty much harmless, but they're also relatively good at surviving in the environment. And so then containment becomes an issue, a proper disposal of the stuff we're working with. So there's lots of experimentation going on, which I don't really know anything about, trying to find different organisms to work with that simultaneously easy to work with and very safe. And it's not something I know a lot about, possibly, so I will mention it. I'm not sure. Next, maybe? As a little tip when I made this, it's a little bit vague. So the safety equipment. Most of modern molecular biology is pretty safe if you're careful. There are some nasty chemicals you don't want to play with too much. There's some power supplies, 200 volts, 400 milliwatts. It's not that scary to most of you guys, I imagine. Centrifuges and various bits of machines can go wrong, but rarely do, unless it's that one. So the free nasty chemicals, they're generally okay. Protect yourself, protect your skin. So if you wear decent gloves, coat, goggles, work in a well-ventilated area, you'll mostly be okay. There are some chemicals you really don't want to inhale. There are some things you really don't want to get in your skin, but for the most part, they can be avoided. And those that can't be avoided, I guess you'll just have to build around them. Yeah. And that's just a burn that bridge when you come to it kind of thing. It's tricky, but yeah. And we work with Bunsen flames, but don't wave your hand through the flame, you'll probably be okay. And the organisms you're working with, there's no escaping the possibility that we might accidentally make something nasty that you don't want to eat, you don't want to ingest. I don't know what to say to that really. Try not to drink it. But quite generally, certainly most of what I do in the lab is pretty safe. There are some chemicals that worry me. There are some things I'm quite paranoid about. But most of that I can't see coming up day to day. And some of the things that are problems along those lines, as I say, we'll just have to do when we get around to it. I mean, there are people here that play with very dangerous equipment. I'm sure anyone who can trust themselves to learn how to use and be safe in sort of a hardware room like we've got next door can learn to operate safely in the lab. It's exactly the same thing. You learn where the hazards are and you're careful. Beyond that, it's not too scary. Finally, right, yes. And now the most fun part of the evening is the bit when I stop talking. So yeah, that's my molecular biology in a nutshell. I will put these slides, I presume the Hackspace can host these somewhere.
Stick on the wiki is fine.
Okay, yeah, so I'll make these slides available for anyone who wants to go back and look at them. And I'm always sort of like uncomfortably enthusiastic about this stuff. So yeah, come and ask me any questions either now or later, as you prefer. And okay, I'll shut and let's start with the interesting bit.
Should we have a quick 5-minute break?
Yeah,
Since we've been sat there for
okay,
an hour.
yeah. So